Every day, we are exposed to a lot of pathogenic microorganisms. To protect our bodies from these organisms, we take different medicines and substances. Vaccines, antibiotics, and insulin are considered highly valuable among the various medicines and substances used for protecting our bodies. Vaccines are designed to provide protection against viral pathogens, such as viruses. Antibiotics are effective against bacterial pathogens, targeting and combating bacteria. Insulin is a medication for diabetes that helps regulate and lower blood glucose levels. All of these products are generated through a scientific method known as biotechnology. Now, the fundamental question arises: what exactly is biotechnology? Biotechnology is a scientific discipline that harnesses biological substrate to create a diverse range of products. That means, biotechnology involves the conversion of substrate into valuable products. And substrate can encompass both organisms and various components or parts of organisms. The products can include vaccines, antibiotics, insulin, and a multitude of other valuable substances. Insulin or INS, and insulin-like growth factor 2 or INS-IGF-2 genes, as well as microorganisms like bacteria and yeast, serve as substrates for the production of human insulin in biotechnology. Recombinant DNA technology, fermentation technology, and downstream processing are key production processes. Human insulin is one of the valuable products obtained through these production processes. Biotechnology is classified into two main types. One is traditional biotechnology, and another is modern biotechnology. Traditional biotechnology primarily encompasses practices such as crop and animal breeding, as well as the utilization of microbes for the production of beverages like beer, wine, and yogurt. It involves various processes, including fermentation, plant breeding, and animal breeding. During the fermentation process, microorganisms are employed to convert substrates into desired products, such as yogurt, bread, and beer. In plant breeding, plants are selectively mated to develop improved varieties with desired traits, such as increased yield or disease resistance. And animal breeding involves selectively mating animals to perpetuate or enhance desirable traits, including productivity or disease resistance. Modern biotechnology entails the utilization of scientific techniques, particularly recombinant DNA technology, to modify and manipulate living organisms for practical purposes, revolutionizing healthcare, agriculture, industry, and the environment. Now, another question arises: what exactly is recombinant DNA technology? Recombinant DNA technology involves the manipulation of DNA from different sources to create novel genetic material with specific traits. Let's know the process of recombinant DNA technology in short. There are several steps involved in recombinant DNA technology. The first step of this process is isolation of DNA and selection of DNA fragment or gene of interest. At first, an organism is selected as the source for extracting the gene of interest that can be a human or another species. Secondly, cells are collected from the selected organism. Then DNA is extracted from the cell's nucleuses. And lastly, the gene of interest is isolated from the cell's DNA using a restriction enzyme. The second step of this process is isolation of vector DNA molecule or plasmid. Unlike other organisms, bacteria, yeast, and archaea contain a small and circular DNA called plasmids. And in this step, this plasmid is isolated from the bacterial cell, which harbors distinct elements such as the origin of replication, selectable marker genes, and a multiple cloning site. The third step involves DNA insertion. The DNA fragment or gene of interest is incorporated into the multiple cloning site of a vector DNA molecule or plasmid. This is achieved by utilizing the DNA ligase enzyme to join the DNA fragment with the vector DNA, resulting in the formation of recombinant DNA. The fourth step is transformation of recombinant plasmid. During this stage, the recombinant DNA molecules is introduced into the host cells such as bacteria or yeast, through a process known as transformation. The organisms that harbor the recombinant DNA are referred to as transformed microbes. The last step is expression of recombinant DNA. In the following step, the transformed microbes are introduced into a growth medium or bioreactor, where they undergo large-scale cultivation. The expression of the transformed microbes in this environment results in the production of a wide range of products, such as insulin. Modern biotechnology encompasses various fields and disciplines. The first field is genetic engineering and synthetic biology, which encompasses the manipulation of genetic material to create recombinant organisms by combining genetic elements from different sources. The second field is pharmaceutical biotechnology, which focuses on the production of pharmaceutical products using various substrates, such as the creation of vaccines derived from viruses. The third field is agricultural biotechnology, which involves the development of crops with enhanced resistance to pesticides and herbicides using various substrates and techniques. The fourth field is industrial biotechnology, which focuses on the large-scale production of various macromolecules, primary and secondary metabolites, enzymes, and other products using fermentation processes. The fifth field is medical biotechnology, which encompasses the diagnosis of various pathogens from different samples using techniques like PCR, polymerase chain reaction, or other advanced processes. Like, share, and subscribe to this channel.